and we're going to have some changes in the program since Professor Dalia Shemien is not coming. So instead, we'll have two major lectures by myself and with Ilya, and then we'll have two students that will take time that Dalia is supposed to have after our talks. It will be Baruch Narodsky and Sivadim Khanati will give their short talks after they won the prize today, okay? All right. My talk is going to be about molecular mechanisms of cigarette smoke-induced muscle catabolism and implication to sarcopenia. And I come from this building here, right here. That's the medical school of the Technion, right on the tip of the Haifa Bay. This is the Haifa Bay. That's the city of Haifa. And you can, local patriotism, that's the beautiful city in Israel. And I live right here on the hill, right across from the bay, and I can see this building from my house. And if you look at the building at night, I can see where my lab is, and my lab is right here on the 7th floor. And I look at the evening with my binoculars, there's no light. And I can tell the student didn't come to work at the evening. Okay, I come the next morning, I ask them, how come nobody works in the evening? Nah. I know you didn't work. But what's interesting about it is that here on the 6th floor, for the last 28 years that I've been in the faculty, there was lights every night. Every night, even today, you go see there are lights there. That's the lab of So now you know why they got the Nobel Prize and I didn't. No, the reason this lesson I take from that is it's not enough if you're brilliant and genius, you have to work hard. And these labs have been working very hard, and the fact that they were the first Nobel Prize in science in Israel, and ever since, every 2 years we get a Nobel Prize, so we're expecting this year another Nobel Prize. I'm see that we'll get it or not. I hope we'll get it. So the idea was that ubiquitin was discovered in our building. The whole story of ubiquitin-mediated protein degradation started in our building, and all the mechanism, how ubiquitin works, and all the 3 enzymes that are involved in ubiquitin, all discovered in Hanover and Hershko laboratory, and for that they got the Nobel. We're going to talk about it because some of the work I'll show you involved with the ubiquitin story in our work. Sarcopenia comes from the Greek word sark, which means flesh, and penia, which means loss. And the loss of lean body, a lean mass or lean tissue. And the definition is the involuntary loss of skeletal muscle mass and strength during aging. And to make it clear, and the point that one picture worth 1,000 words, just look at this guy who happened to be Maolam, the strongest man in the world in the 70s. And sarcopenia means changing from this into this. Same guy. Okay? But look, if you know a little anatomy, look at the pectoralis major muscles. Look at it there today. Oh, that was in the when was the governor of California? 50 years ago? Look at the muscles of the rectus abdominis. Doesn't matter if he was the strongest man in the world, he is sarcopenic. Look at this guy. Look at the muscles here. He is 18 years old. Look at this is the quadriceps muscle. These are the gastrocnemius, huge muscles. 18 years old. You don't want to look at him today. It's me when I was 18. Okay, we lose muscle with age. It's very hard to do something about it, but we'll show you today that we start trying to understand why we are losing muscles and what's the reason for losing muscle, how the molecular mechanisms are. Okay, that was in the Sachne, by the way, if you want to know where the picture was taken. So we divide the factors that are involved with sarcopenia to intrinsic and extrinsic factors. The intrinsic factors are basically a few categories, like inflammatory activity goes up, and the apoptosis goes up, which will lead to muscle fiber loss and atrophy. Oxidative stress, we'll talk about it quite extensively about it. And by the way, there is an oxidative, Israeli Oxygen and Free Radical Society that I was happy to be a president for a few years, and you're all invited to join. The oxidative stress causes the upregulation of MEP kinases and NF-kappa B pathways, which lead to ubiquitin proteasome increase, which will lead to protein degradation, protein breakdown, which of course will lead to muscle fiber loss and atrophy. And anabolic hormone goes down with age. The result is protein synthesis goes down with age. When protein synthesis goes down and protein binding goes up, of course, you'll lose muscle. The extrinsic factors have to do with way of life. Depends what we eat, impaired nutrition, depends whether we do exercise or we don't exercise. Most aging people reduce physical activity, which will lead to immobility, illnesses. Catabolic medications will cause muscle breakdown. But the thing that we have been concentrating the last few years is whether smoking may lead to muscle breakdown, and especially in old age. Cigarette smoking is still a problem. 20% of the world population is still smoking. So it means that 1.5 billion people, we say in Hebrew 1.5 million, out of 7.5 billion people that are on the globe today, 20% still smoke today, and in some places 40%. In China, it's 40%. Twice as the average, okay? In China, it's a massive thing. Cigarette smoke is probably the single most significant source of toxic chemical exposure to humans. And finally, there are over 5,000, today we know close to 6,000 chemicals constituents in cigarettes. So, the problem of smoking is not just the active smoking. If you are near a person like that, half a meter from you, and you see all this smoke around, you're a passive smoker. And why a passive smoker is having even more problem than an active smoker? Because he inhales smoke with no filter. If I get this smoke into my lungs, it didn't pass any filter. While this guy still have a filter. So passive smoking, in a sense, you get much less smoke, but you get 10 times more of the toxic chemicals when you smoke as a passive smoker. By the way, who is it? Anybody know who he was? We are the oldest, right? The young guys don't know. So now let's tie the idea of sarcopenia and smoking. Smoking has been shown to be associated with sarcopenia in previous studies, and from epidemiological studies, smoking was identified as a risk factor for sarcopenia. And in the early of this 21st century, some studies involving thousands of people, hundreds of people, thousands of people, showed that you can correlate the fact that heavy smoker and aged or older people are having faster sarcopenia than normal people who don't smoke. So if you're a heavy smoker, you're going to lose your muscle much faster than a person who doesn't smoke in old age. So that was epidemiological studies. But if you go to clinical studies, okay, taking biopsies from smokers versus non-smokers, you find out that there is atrophy of oxidative muscle fibers, especially. You find out the decreased rate of muscle protein synthesis and overexpression of autogene 1, one of the E3s of ubiquitin system. And that was also papers that were done in the early of this century. And finally, work that I've now been doing in vivo and in vitro studies on animals and muscle culture cells show muscle wasting, downregulation of myosin heavy chain, expression of USP19 protein that dissociates ubiquitin, and activation of P38 and MEP kinase systems. So we developed in our lab many years ago an in vitro system of smoking, which means that we can smoke very sharply, quantitatively, and reproducibly the same amount of smoke every time we want. And how it works? It works by taking a chamber, putting in some kind of closed system, and hooking up to a vacuum pump. And the vacuum pump is controlled by a barometer that you can measure exactly the negative pressure that you form inside this vessel, whatever you call it. Now, so we have two valves, valve A and valve B. Valve B is closed, and we open valve A and turn on the vacuum pump, and we can measure the pressure here. And we usually can do any kind of pressure, and we can show later on that when we close valve A and we light the cigarette, okay, and open valve B, by the vacuum that was formed here, the negative pressure, we can pull in the smoke in a very quantitative amount. Therefore, we can, by deciding what pressure we want, we can decide what kind of amount of cigarette smoke will go into the chamber, and here we can put our muscle cells, whatever, or muscle fibers, or whatever you want, any kind of biological systems, where we can quantitatively get smoke and then incubate it for any times we want in order to see the effect of the smoke on the biological systems. Is that clear? Very simple system. Very simple and reproducible, because we can measure from the smoke some parameters that come in and correlate it with the pressure in a very straight line. Control is just putting to the system air exposure. Sham air exposure is our control, exactly the same thing, no secret. So we took myoblast, differentiated it into myotubes. And myotubes look like myosin fibers. Or not only that, when they differentiate, they express myosin, while here you don't see any myosin. And they start contracting. I don't have the picture here, but we have movies to show how they start contracting. And how do they contract it suddenly, simultaneously, and spontaneously? Anybody knows why muscle contracts spontaneously? What does make a muscle pain?
Iron concentration.
Right. Iron concentration of calcium. Calcium concentration, when it goes above certain level, certain, it causes the muscle to contract. So now we go and we smoke our fibers, or okay, our molecules. And we do the following. We smoke it, and we ask the question, does oxidative stress involved in this system? So there's a very known assay called the DCF assay, dichlorofluorescent assay, which basically is a fluorescent assay that after this chemical goes into the cell, it becomes fluorescent when it's oxidized. When there is oxidative stress, it becomes fluorescent. And if there's no oxidative stress, no fluorescent. So you see, after 20 minutes of incubation of one exposure of cigarette smoke, 10 seconds of cigarette smoke exposure, and incubation for 20 minutes, in a minimum pressure that we can get, which is 50 millimeter mercury, we can see a huge increase in oxidative stress. But if you put N-acetylcysteine right here, which is a known antioxidant that goes into the cell and reduces the oxidative stress, we go back to the control level. So the first thing that we already know, and we know that from many other studies, the cigarette smoke causes oxidative stress in any system it comes, because there's a lot of oxidants in cigarettes. We look at the MET kinases, and especially the P38 MET kinase activation and NF-kappa B activation by cigarette smoke. And you can see that as a function of time, under 50 millimeter of mercury, the minimum, the smallest amount of pressure we can get into the system, the negative pressure, we can see activation of the P38 by phosphorylation of the period compared to the unphosphorylated, which hasn't changed. So we activated the P38 MET kinase. We activated the I-kappa system by phosphorylation of it and degradation of I-kappa, which is the inhibitor of NF-kappa. And also the P65 major component of NF-kappa B is being degraded, it disappeared from the cytoplasm into the nucleus, and that's how NF-kappa B basically started its transcription. We went to look at the ubiquitin system, the famous ubiquitin. And ubiquitin system was discovered that works very, very complicated. It's not just you hook up ubiquitin to a protein. You need 3 enzymes. It's called the E1, E2, and E3. E1 is called the ubiquitin activating enzyme. It's a single enzyme, one enzyme. that puts ATP on top of the system in order to get it charged with energy. And after that comes two other two enzymes called the E2 and E3, ubiquitin conjugating and ubiquitin ligases. And the function of these two is to give the system the specificity of ubiquitin to how it will be hooked up to different proteins. There are about something like close to 100 E2s nowadays and about close to 900 E3s. And the combination between E2 and E3s gives you the specificity which protein will be hooked up to which to ubiquitin. In the muscle, we find the major two E3s called the atogene 1 and MERF1. And they are the predominant, there are 5 of them, but these two are predominant in muscle. And they are usually activated or are formed under condition of muscle degradation. Otherwise, you don't see much activity of these enzymes. So we ask the question, can we see activation of the E3 ubiquitins, E3 enzymes under the condition of cigarette smoke? So you see how it happens. E2 is hooked up to ubiquitin, E3 usually brings the protein, and the specificity between them will tell which protein will be hooked up to the ubiquitin. The ubiquitin will be taken with the protein into the proteasome and be degraded in the proteasome. So if we looked at the results, we saw very clearly that the messenger RNA, by real-time quantitative PCR, you can see that within 1 hour, you get an increase about almost 100% in the messenger RNA of both MERF1 and atogene 1, and in 2-3 hours it goes down. On the protein level, of course, it goes much farther and goes longer, and by 6 and 12 hours you see a very large increase, something like almost 8 times more of the level of these two E3s of ubiquitin in our muscles as a function of cigarette smoke incubation. And going a little further, other proteins in the muscle being degraded. Can cigarette smoke cause degradation of muscle protein? So the two major proteins in the muscle, myosin heavy chain and actin, as a function of time of incubation to a single puff of cigarette. And you can see how it goes down. As a matter of fact, by 6 hours, by 12 hours, myosin can almost completely disappear. And actin, by 6 hours a little bit, usually by 6 hours you don't see much effect. By 12 and 20 hours, already very large effect, which is statistically significant. So here's muscle protein are being degraded just by incubation with cigarette smoke. So we go farther than that. We go to the level, to morphological level. What happened to those myotubes? And we look at the diameter. Okay, cigarette smoke induced reduction in myotube diameters. And look at the diameters control after 1 hour of incubation and after 12-hour incubation. The myotubes are shrinking. The proteins are breaking down, the myotubes are shrinking. And the quantitative analysis of that shows that they're shrinking up to about 50% of their original diameter. That's the original diameter, and after 12 hours you get to 50%, and you stop around 50%. Okay? But they are shrinking just about to half of their original size. So we came up now with a scheme that we try to understand molecularly how cigarette smoke will eventually cause protein degradation, which may lead to sarcopenia. Cigarette smoke and the components of cigarette smoke will obviously cause oxidative stress. Oxidative stress has been known a factor in turning up the MEP kinase, AP38, MEP kinase A and NF-kappa B. Those two systems are affected by oxidative stress. NF-kappa B has target genes of atogene and MERF1, the two E3 ligases ubiquity. The target genes, among many other target genes, it will upregulate the messenger RNA of atogene and MERF, and this will lead to myosin and actin protein degradation, which may lead to sarcopenia. So that was our scheme, and we asked the question, how can we prove it? And we prove it by using specific inhibitors of every one of the steps. N-acetylcysteine, I already showed you, that can inhibit the effect of cigarette smoke. I'll show you more data in a minute. SB is a known inhibitor, specific inhibitor of P38. IMD is a known inhibitor of IKK, the kinase that phosphorylate I-kappa B. I won't show you data, but we have data to show that leucine is an inhibitor of the messenger RNA, of production of messenger RNA, Avraham and MERF1. And MG132 is an inhibitor of protein degradation of the proteasome. Okay? So we can, every one of the steps, put a specific inhibitor, see what's happening downstream. So let's see some of those examples. For example, if I take the effect of NEC and look at the messenger RNA of the two E3s, the MERF and atrogene, I can see that within 1 hour it goes about almost 2 times more, but under NEC it goes back to control level. So I can show that indeed I do not upregulate the E3s of ubiquity. Under protein level, the same thing. This is MERF under protein level, and you see it goes up by 6 hours, but under NEC it doesn't go. NEC reduces the expression of the protein after 6 hours. Myosin heavy chain is degraded very nicely after 6 hours, but with NEC it goes back to the original, very close to the original of the level of the control. Same thing when we take the SB. We look at the messenger of the E3 ubiquity enzymes, and you see exactly the same thing. It goes up with 1 hour, and SB completely brings it down to the level of the control. Protein level, the same, it goes up in 6 hours, completely disappears under SB conditions. Myosin heavy chain is degraded very well, but it's not degraded when you put the inhibitor for P38. And similarly, I do with IMD, which is the inhibitor of IKK in NF-kappa B system. Under messenger RNA, you see there is good activation after 1 hour of the messenger of the 3E3 ligase. But here the IMD specifically inhibits the MERF1 and not atrogene. And we know why, because atrogene goes to P38 directly and not to NF-kappa B. But when you look at the same thing on the protein level, you get exactly the same results. You look at the myosin heavy chain level, exactly the same results. And if you look at the diameter of the myotubes, again, diameter goes down to 50%, and IMD can attenuate this decrease by putting up again to about 80%. So going back to our scheme, what we are saying is that basically cigarette smoke with its components, that we talk about them, can cause oxidative stress, which will turn on the single transaction systems of MEP kinases and IKK and NF-kappa B, which will turn on the genes of atrogene and MERF1, which will cause the upregulation and the degradation of muscle protein, which will lead to sarcopenia. That's the scheme that we made up, and we published it in about 3 or 4 papers in the last few months that have come up with this molecular mechanism. One of the interesting story was what ingredients in cigarette smoke may be responsible for muscle degradation. It's not only that that we want to know the mechanism, we want to know who's doing it. And in the cigarette smoke, there's a lot of potential chemicals that can do that, something like RNS and ROS, reacting oxygen, reacting nitrogen species. And we know that they can do it, but in vivo, it's very difficult to see how they survive because their half-life is very, very short. If you take cigarette smoke, the RNS and ROS hardly will get into your blood system. But aldehydes from the cigarette smoke, especially acrolein, which is a very active unsaturated aldehyde, and also acetaldehyde, these are the major ones that exist, and people have measured them under cigarette smoke, and blood levels go up. So we decided to look at them. And if you look at acetaldehyde, and you look at the data, you saw that acetaldehyde didn't do very much. It did not change the level of myosin heavy chain, it did not change the actin, it didn't change the atrogene, it didn't change the MERF. So our conclusion was that acetaldehyde is not doing any degradation. It does do slowdown of protein synthesis in alcoholic myopathy. Alcoholic myopathy, which alcohol forms acetaldehyde, you will see that in people who drink a lot of alcohol, they have alcohol myopathy due to the slowdown of synthesis, but not degradation of muscle. But acrolein is responsible. Now, we found out that acrolein is the guy mostly doing a lot of degradation. If you put those myotubes in under acrolein, you see increase in the E3s. You see increase in the E3s on the protein level. You see increase or degradation of myosin heavy chain under acrolein condition and acrolein incubation, and you see the degradation mainly of myosin under this condition. So our conclusion was that acrolein is probably the main component of cigarette smoke that is responsible for cigarette smoke associated muscle catabolism. To end, I want to, of course, to give the credit to Owen Rom and Sharon Kisari, who did the work, the actual work of this project. These are the people in the lab last year. And the fact is that they are the average age, they about 30-something, 31, 32. And I'm the only one who's getting old in the lab because the average age in the lab stays for the last 30 years the same. The only one who gets old in the lab is me, okay? So that may be the reason I will stay forever young, maybe. Thank you very much. And then, if you don't why the global warming, just look at the panties. Thank you. Questions? Yeah. That's all we need.
What kind of cigarettes did you use?
Time cigarettes. We use all the time cigarettes. We know exactly.
Very nice.
And we don't use cans or something like that. We do little local cigarettes.
Second question: concentration of smoking materials. Can
We
you
measure them.
compare it to what we have?
Yeah. Okay. All right. Good question. How we sort of compare it to the amount of cigarette smoke that we take in, right? That's what you're asking. We can do it by acrolein, and what we measure as kind of gold standard of how much smoke goes in is nitrites. Cigarette smoke causes nitrite because there's NO. NO makes nitrite, and you can measure the amount of nitrite. So we know how much smoke brings how much NO and how much nitrite are formed. So we take the amount that usually equivalent to one cigarette. And that's where the puff comes in. Although it's one puff, but we incubate it a long time, so we fall in more and more nitrite. So I didn't show you, but we know how much nitrite, and from that we can quantify how much cigarette smoke and how it relates to the smoke of one cigarette. Yes?
So you're showing accelerated sarcopenia, basically by smoking. Is it related to the natural sarcopenia? Are you?
No.
So this is.
The natural sarcopenia is much more complicated than that because there's no chemicals. It has to do with a lot of things. As I showed you before, it has to do with physical activity, with nutrition, general nutrition. It has to do with a lot of way of life habits and things like that. And also, I believe in genetics.
Okay.
Some people will have minimal sarcopenia, and some people have terrible sarcopenia and nothing to do with cigarette smoke. And we know that there is a genetic factor there too. So we don't know which genes, but we know the genes are involved also in muscle breakdown, some certain genes.
On top of what you just said, wonderful talk, by the way.
Thank you. You heard it the third time in the last half year.
And there is, anyway.
I know, this is the third time
But
you
you improve every
And the
time.
last time you went away in the middle of the lecture.
I know what's
Next
going
time
to
he will give it.
But there is one question that is, in my thought. This is a good presentation for the algorithm tomorrow. How this presentation associated with aging? It means you don't have to smoke, but what?
Right. Absolutely, this is a good question. What I showed you here is the effect of cigarette smoke on muscle breakdown. With aging, it's much more complicated. For example, the people who exercise, they're slowing sarcopenia. Exercise is a good thing against sarcopenia. For example, we showed data, leucine, I take every morning a lot of leucine. Because we know that leucine activates the E3 ubiquitin, inhibits the E3 and the ubiquitin system. So I just came back home, right, as you know, last week, and I went to a nature store and got leucine. And it says there that it stops muscle breakdown. They know it in America for many years. So I would go say, people, if you want to have good muscle, take a lot of leucine, it works. It works in our system, it works in all over the United States, in many, many studies. There are ways to slow down in aging, nothing to do with cigarettes. Taking leucine, do the proper exercise, you slow down. You cannot avoid it, but you can slow down. By the way, we lose 30 to 40% of our muscles after 65 years old. That's a lot of muscle. A person who weighs 70 kilos has 28 kilos of muscle. Okay, 40% of our body is muscles. Now lose out of it 30, 40%. But at the time, you don't lose weight because you're gaining fat when you lose muscle. And that's very bad for aging because when you don't have strong muscle, you fall, you break the leg, you break the bones. So muscle are very important.
But you didn't answer my question. My question was, what in your study mimic the environment components that affect aging to have sarcopenia?
Not in this specific study.
So the smoking study is not
really
aging
aging, but
not
you can
aging
do it.
related.
No, but you can do
Thank
it.
you.
All right. It does explain, I think, that the molecular mechanism will be also similar in aging.
Yeah, but there's a lot of components that drive the whole thing.
Right.
Yes,
It will be masking that. Right, absolutely.
This molecule can be organic from the apple. I think without it, the damage is not happening.
No, when you, it can imitate the cigarette smoke, but if you use along the whole scheme that I came, you can stop it by individual inhibitors of very one of the steps. You can do it in cigarette smoke, you can do it with acrolyte. Yeah. Oh yeah, there is. You can get most of the acrolyte out, 99%. If you put what's called active carbon, you write everybody you write active carbon. You know, active carbon, Pahman Payil. If you put in the filter Pahman Payil, what we call active carbon, most of the acrolyte doesn't go through. When the cigarette smoke companies try to do it, they're starting losing money. Why? Because the smoke that comes out without acrolyte is worth nothing. Most of the smell comes from acrolyte. And people don't want to have a smoke, which is smoke, no taste, no smell, no nothing. Okay? So they need acrolyte. So that's a problem. They cannot get rid of the acrolyte. Yes.
Maybe it's more suggestion, not a question. If smoking promotes protein degradation, we know that there are accumulation of damaging proteins in it. So maybe smoking could help to degrade
That's right. Damaged protein, because they're damaged, the ubiquitin system does not recognize. The problem is that the whole idea of ubiquitin, about 70% of our muscles, not all of them, are degraded by ubiquitin proteasome system. Okay? But the system is very specific. It has to recognize specific protein to be degraded. It's not a general thing that every protein that is not damaged, the proteins, the ubiquitin system will automatically degrade. It doesn't work that way. So because it's a specific system, your idea is not so simple. Yes.
Also the data that you showed, is in tissue type. So how much of the data shows that in the smoke pass through the lungs to the blood and directly to the muscle? And if you ever did experiments with
No, but people have done in mice the same experiment. They took mice and smoked them for a long period of time, and they looked at what happened to the muscle, and they showed that the mice that have been exposed to smoke for most of their life. They have very, very bad muscles compared to mice that eat. So we have in vivo or perfect that does better. Okay, thank you. The next one, the next lecture is by Dr. Ilya Tendler, Application of Information Theory.